Hello, Canada. I am very glad to be here. Every year I come about this time. Josh, is that right? And it's minus 15 degrees. Well, what's with the heat wave? That's what it's a real pleasant switch. Um, what I want to do today is get through quite a bit of a transitional thinking. You know, we talk about soil health. I think that was maybe the first two words up there. You'll hear bacteria, you'll hear fungi, you'll hear all sorts of things. But it's really the bigger picture we want to look at. And you'll see a definitive switch from just talking about adding bacteria or adding fungi to adding useful functions into the soil. And it's really the theme. And if there's anything to take from this product science presentation, it's what functions can we add into our soil to make it healthy for the crops and to promote the biodiversity of the whole system there. So as we go into that, of course, we're going to start talking about the rhizosphere, which is that little narrow zone right at the root soil interface where you have plant exudations, you have bacteria, you have fungi. It's a real complicated thing. We all know and have heard the term, and it's really important to have it right, but we're not sure what that means. So all kinds of researchers have been looking at plant growth promoting bacteria or plant growth promoting fungi. And I'm just going to, as we go on, just focus on plant growth promotion. But it's been going on for a couple of decades now. And, as we do in academia, sometimes you can't change 20 variables and get an answer. So a lot of times, and most of the research, whether it's fungal or bacterial, it's with a single species and they're looking at, very defined things. I think it's important for us in the practical world to understand, take any aerial view of a crop field. And if you see something that looks homogeneous, all the same, you're not using your eyes. It's never that way. So there's so many differences, even from point A to point B within the same field where the soil's different, the microbiology is different. So, a single strain going into a given field, does it really pass the smell test? And I think the answer is it really does not. Having said all that, we do disclose, just as Travis pointed out, we have these 5 species of bacteria in the ACF main formula. For one, these are all on the Canadian DSL, the domestic substances list, where you can bring them into the country. And we have one Rhodopseudomonas that's a photosynthetic that has ability to work with oxygen, without oxygen, with light, without light. It fixes nitrogen, it fixes carbon dioxide, it breaks down all kinds of things. So it's a workhorse, and it's involved in a lot of cutting edge research. It's maybe looked at something that will help with greenhouse gas emissions and turning atmospheric CO2 into something more useful. But along with that, we have 2 bacillus species. That's a Lecaniformis and a subtilis. We have 2 nitrifying organisms. Having said all that, and those are all present, identifiable, and clearly cut, the species really aren't that important, which is going to sound crazy, but it really, as we go through this, you'll understand. Whether it's plant growth promoting bacteria or fungi, and you look for any summary paper, you'll find that there are several recognized obvious ways that the microbes can help the plants. And for direct plant growth, we have nitrogen fixation, pulling nitrogen out of the atmosphere and making it into an organic form that the plants can use. Phosphorus solubilization, very important. We'll talk about that along a similar line. Potassium solubilization. Siderophores are small molecules that can chelate iron and bring it into the plant. Phytohormones, they're plant growth hormones of various types. Then we have all sorts of indirect plant growth promoting functions, sometimes antibiotics, hydrolytic enzymes we'll talk about quite a bit because These are what take complex organic materials and break them down into simple sugars or simple amino acids, etc. And it turns out that whether you're a bacterium or a fungal organism, you have to have the real simple stuff. Otherwise, you can't eat it. So that the hydrolytic enzyme is very important. And there's all kinds of documentation of improved disease resistance and so forth. Now, that's starting to make the transition to functions. And that's, again, really what I want to stress in this presentation. When we listed the direct functions, the first one we listed was nitrogen fixation. This is electron micrograph of Rhodosudomonas with its red pigmentation is a very potent nitrogen fixing organisms. You'll see as we go through this, for any of these species that we have, we can take the identical species and grow it special ways, either excluding some nutrients or providing it with what are called inducer compounds, things that we can do to make sure that these important functions that we just saw in the chart, that the organisms have that built in so that when they are added to the brew systems or directly to the field, they provide those functions. And it's really, really the key to everything. So the Rhodosudomonas does a great job of fixing nitrogen. The second main function on there, and we talk about this a lot today, is phosphorus solubilization. Any of the organisms we're talking about, plants, bacteria, fungi, they really need orthophosphate. And I think we've all heard these terms a bit. You have bioavailable phosphorus and you have non-available phosphorus. And the orthophosphate's the form it's got to be in. A lot of times in soil, most of the phosphorus is locked up in a non-available form. We have a specific slide on that. But making sure that the culture has the function of being able to convert non-useful phosphorus into bioavailable phosphorus is one of the most important things that we make sure we keep in the product. Just some examples of how you can do it. This happens to be a Bacillus subtilis. The Petri dish on the right side of the chart there, that has tricalcium phosphate, TCP, in the plate media. And it's not soluble, it's insoluble, it looks like chalk when you have a pure supply of it. And when you grow the organism on it without other phosphorus available, it can dissolve the TCP. And when it does that, it has that little halo or clear zone around. You see the actual colony in the very center, and you got a halo around it. And the size of the halo is directly proportional to how well that colony is breaking down the non-available into bioavailable phosphorus. So we have about 1 million tricks like this figured out over the last 30 years, but they're all important. I'm only going to give you a few examples. We also listed, we talked about the Rhodosudomonas there, and then we talked about the Bacillus, the nitrifying bacteria. I think everybody knows it turns ammonia into nitrite, into nitrate. One of the things that we found is that there's also some plant growth promoting properties with this, with the nitrifiers, and it helps with the phosphorus solubilization as well. And we'll talk about that a little bit more in a minute. This is a really key chart. You've probably seen this in your old agronomy textbooks. P fixed in the soil, what that means, it's not available. You can't use it. When you have acidic soils, you get aluminum and iron precipitates. Phosphorus is bound that way. When you have alkaline soils, you get calcium precipitates. And in either case, it's not in available form. So you can add as much phosphorus as you want into a really acidic or a really alkaline field. And even if you're adding orthophosphate, the most expensive form, it'll get bound up pretty quickly and not really be available. So, this is one of the things that we're really trying to overcome. You see a little narrow band there where it's soluble, where it won't bind so much, and that's at a pretty narrow pH zone. So it's a real limitation that we have in our soils. Now, in the ACF products, we can take any of those species and, again, grow them certain ways. And we do a lot of things, such as growing them purely on TCP, tricalcium phosphate, with almost no orthophosphate in there, and force them to make the volatile acids and so forth that digest the insoluble phosphorus. And we then put that into the entire consortium of organisms that are come in a jug that then go into the brew tank. And these zones, it's just a visual way to understand it. That's insoluble TCP on the outside. The colonies are in the center. That zone over time at a fixed temperature, that's directly proportional to how well that organism is breaking down non-available P into available P. And those are, tricks like that are what give us the ability to ensure that we're putting the functions, not so much the species, but the functions into the product. Just real quickly, one of the synergies that we have is we talked about these hydrolytic enzymes. If you have proteins or whatnot in the soil, the bacillus can break those things down, liberate ammonia. When ammonia is present, not only can, in early, just post-germination, that ammonia is taken up by the plants, but then the nitrifiers can convert it into nitrate. And in the process, the nitrifiers are enhanced at breaking down and solubilizing P. So that's sort of a little cycle that we have. The whole thing works really well. But, there are many species that can do that, but we make sure that we have a defined rate at which we can break down the insoluble phosphorus and make it bioavailable. So we're adding that function into the soil. So having said all that, here's what we consider our function fingerprint. As we look down in the very first column, we have a bunch of important functions. And then going across, we have the 5 species that we talked about. And you notice some of them do different things. For example, the first one, the subtilis, is really good at making protein digesting enzymes, and it's pretty good as we go down with more plus signs. And, all of that can be done, but only using, all these tools that I briefly illustrated with those zones of clearing on the phosphorus. So this functional fingerprint, the transition we need to make when we're thinking about what we're adding in terms of a probiotic to crops. Does it matter if we are adding the species, if they grow or not grow? That's not so important. What's important is that we're adding these specific functions into the soil. And we will talk about why that's important based on this type of chart. While fungi reproduce differently, bacteria, they split by fission. 1 turns into 2 to 4 to 8 to 16, etc. And this is just an example. The center column in the chart, that's bacteria that reproduce every 30 minutes. And then on the right-hand side, those are bacteria that reproduce every hour. After just 5 hours, you have 1,024 of the fast growers, and you got 32 of the slow growers. Now, you can understand in any mixed food environment, whether it's on the field or in a beer fermenter, anything that by species that's growing faster is going to quickly dominate. Here in this situation, you got 0.3% of the slow guys, and you can imagine you take it out a few more hours. It's really easy for the slow reproducing organisms to get lost. And it's just a matter of who's going to grow the fastest, and it really is literally the survival of the fastest. And what we want to talk about is all these different functions, all the different efforts that we put into making sure these bacteria can make protein degrading enzymes and chitinases and plant growth hormones. While they are doing those things, they're using the material and energy that they have to produce those products, those functions, and their reproductive rate slows down. It's literally just by definition, if you're making things that aren't directly related to your reproduction rate is down because you only got so much material and energy left. So without a doubt, when you have a culture that is producing all these additives and the functions and the enzymes and everything else that we showed in our fingerprint chart, their reproduction rate is low, and that's unavoidable. And the organisms that are just growing and reproducing without doing that stuff, their growth rate is fast. So we just saw 1,024 versus 32 in 5 hours. So, we can all do that math. Now, who or what benefits from the supply of all these plant growth promoting functions? And now we're really making the transition from talking about bacteria or fungi to these functions. When you add these functions into the crop, you're not really adding the species that are generated in that brew tank. I mean, we know what they are, we can count them, but what we're really adding is the functions. And those functions, they provide extra nutrients, whether it's simpler organic compounds or fixed nitrogen or more available P. Who or what is going to take advantage of that? Well, it's going to be anything that's in a location where those nutrients are supplied. So all of those functions, including the P solubilization, the growth hormones, the entire biomass benefits from that. And it's certainly not, just the organisms that we're putting in there. And, we've done a lot of studies of tracking the populations of different things over time, post inoculation. And invariably what you find is that we see the numbers of what we put in because we put it there, you can measure it. But the growth of the other organisms goes up dramatically. Why? Because those promoting functions boost the nutrients, the hormones, the increased available P, et cetera, for everything that's in the biomass. So this is never a case of adding our bacteria and they take over the crop or they take over the ground. It just doesn't work that way. We're literally putting in functions rather than numbers of bacteria. So our real advantage is that anytime organisms are grown as fast as you can in a molasses broth or anything like that, or compost tea, and people are just trying to boost bacterial numbers or fungal numbers, all that you get are organisms that can grow fast. They are not necessarily producing any of the useful functions. They're not really delivering anything. You can have 10 to the 9th or 10 to the 13th. It doesn't matter if they're not doing the things that break down the phosphorus, that fix the nitrogen, that break down the complex organics. And the fast-growing organisms don't do a good job of that. So, we recognize that we've spent literally 30 years figuring out how to confer enhanced functions on the culture and then how to take it into the brew tank, which we're anxious to unveil, and make it economical and predictable for everybody. Having said all that, I do want to say a little bit about fungi and bacteria. You hear a lot of people, and I think Joshua alluded to it earlier, that you have, well, I have trichoderma, you don't have that, or we have, whatever count. What does all of that mean? So I'm just going to compare and contrast fungi and bacteria for a bit here. Bacteria on the left, this is not to scale. Bacteria are really small. fungus are larger and some occupy entire forests. Uh, you have bacteria were around about 3.5 billion years ago, as opposed to about 1 billion years for fungi. We already talked about the reproduction. Bacteria split in two into four. Fungi have both sexual and asexual growth, and a very different situation there. One of the things that's very important is this protection against the environment. A cell wall is something that surrounds all bacteria. It's a semi-permeable membrane. It prevents complex stuff from getting inside, so the bacteria have to have simple stuff to eat. And fungi, as far as complex organisms, they're the only complex organism that has a cell wall. And that's also important because just like bacteria, fungi have to have simple soluble material as a nutrient source. Just to give you an example of what we're talking about, the major protein in milk, casein, is too large to pass through the cell wall of either the bacteria or the fungi. And that casein has to be broken down into its constituent amino acids before these guys can eat it. So even something that you might think is soluble, like milk, is not for the purpose of these organisms. They have to have the simple soluble materials. Another factor is, I've seen some good results from adding fungal products, but having said that, they're all supplied, generally speaking, in spore form. Spores have to germinate. What allows spores to germinate? Well, it's a complex variety of things, but for one, you have to have nutrition, which means you have to have the soluble organic compounds. And then there are all kinds of other conditions for specific fungi, but it's not super easy. The mode of nutrition, just like bacteria, is absorption. You have to have the simple soluble stuff or the fungi can't touch it. It has to get through the cell wall. It has to be soluble. So both bacteria and fungi can make these enzymes inside themselves that they excrete into the media, and that breaks down to complex stuff, and that gives them some soluble food. That's the only way they can grow. And as we talked about before, making these enzymes, it doesn't help that organism grow. It slows down their growth. So, it's not occurring in the soil at the rate that would be optimal, whether you're germinating fungal spores or whatever reason. So fungi can make the exoenzymes. When do they do this? They really do it as they go past germination and they form hyphae, which once you get to that point, then they can make the enzymes. But if you're adding just fungal spores, there's no guarantee that they're going to germinate. And if they don't germinate and start to grow, they're never going to make these enzymes. You sort of run into a dead wall. So, and it's never the case that only bacteria or only fungi or only one or two of those functions should be added. It's always a big picture and always a combination of different things. But I'm just pointing out that you can't just take fungus spores and throw it on a field and necessarily expect anything good to happen. All these words here, I'm not going to read those, but the middle statement in bold, the functions we add are much more important than the species or the numbers. You see all sorts of things about how many CFU or, this, none of that, as far as we're concerned and as far as we've demonstrated, that doesn't mean a thing. It's how much activity of the right functions are you adding and at what cost. Here in Western Canada, it's almost all the soil is alkaline to one extent or another. And when you have that type of thing, it has an impact not only on the phosphorus availability, but on fungal growth as well. And this is just to put everything into context. We have this picture. This is a trichoderma species at pH. And this was done in a series of plots across New York State. And as you can see, when you, at the far left, you're at pH 3 and you get up to pH 9 at the far right, almost all the fungi, certainly the trichoderma, they have really good growth at a high pH. When you have, forest type soils and you have an acidic pH, or you have a high fungal population, when you have alkaline soils, the fungal population never really gets up there. And so these sorts of things, it all comes into play when you're talking about, do you want to add bacteria or fungi? And remember, what we say is none of that matters. You really got to add the functions and let the natural biomass take advantage of that. We're going to talk a little bit about something that's coming down the pike very, very soon. For the last several years, we've been working very hard on biopesticides that, for one, have some of the plant growth promotion properties, but for two, are really strong producers of an enzyme that breaks down the fungal cell wall, which is largely made of chitin, and it's called chitinase, the enzyme that's being produced. Trade name, chitotrol, a non-toxic biopesticide. There have been some spotty trials, nothing that was significant here in Canada, but people have seen some real good results with that. There's been no sale yet because it's not legal, but there are countries where, we've been able to get it out there pretty well. What we have here is just a diagram, and it's just for emphasis, so we don't have to worry about all the chemistry going on, but the cell wall is largely made of chitin in fungal organisms. And by disrupting that cell wall, you can attack a fungal infection. A really interesting project that we're now equipped with the efficacy data, so that we're starting now officially with the US EPA and its counterparts in Canada to get biopesticide registration for the chitotrol product. Black sigatoka is a fungal infection of bananas that literally stopped commercial production of bananas in Florida and is a real problem elsewhere. In a project in the Dominican, which was run by one of their best agronomy schools, we compared the best practice there, which is a copper-based fungicide, Phyton 27. I think it's sold here in Canada. Chitotrol that's not brewed, that doesn't go through the tanks, and T3 activated chitotrol, that means brewed. It goes through a brew process. And what we have here is the standard practice has an index called the evolutionary development of the disease, and they look at leaf eruption rates and visual things, but an index of 200 means apply more fungicide right away. In this particular case, this was started when all three control and the two chitotrol tests were started when the disease was pretty severe. After one dose, you see the blue is the Phyton 27. It never really gained significant traction. And again, the researchers started with a pretty high infection. As we went there, the unbrewed powdered chitotrol did a decent job. It took it below the threshold for applying fungicide, and the brewed chitotrol, which we'll explain, the tanks, did a fantastic job. So the big thing there is brewing. And fortunately, back in the States, we like to do things sloppy and dirty. We don't have whatever the ingenuity to set up good brew stations. We just make bacteria perform functions or whatever. But the idea, and we don't have to worry about the chart too much, is you take the right mix, and it's a very defined mix of bacteria, nutrients, the amount of water for the concentration, aeration, temperature control, and you brew it for a period of time that forces the culture to do something in particular. Yes, it's reproducing, but then it's producing one of the functions. In this case, it's one of those hydrolytic enzymes. So the brewing process is integral to optimizing the function that we provide, and it is also integral to economy so that we can make things at a reasonable cost. Which brings me to a huge innovation here. Ryan Parker, who's been with Advanced Ag for 3 years, looked at our American brew tanks and was not impressed. So over these last 3 years, he's done an incredible job working with some local engineers, and I think he got some funding too, right, Josh? And came up with something that's really dependable, that gives some real-time telemetry, that automates the brewing process and takes things from hoping it's done to guaranteeing everything's on spec.